Right, so this is a re-record of some of the concepts that I did on Tuesday, during Tuesday's lesson, just to make sure that you've got all the definitions and all the concepts correct. Okay, so remember that under normal circumstances, chromosomes are single-stranded. Okay, and so under normal circumstances, in other words, when a cell is not dividing, every chromosome consists of one molecule of DNA. But remember that that is wound around the histone proteins every now and again. But it's one DNA molecule. And this consists of a series of genes. And between those genes is areas of non-coding DNA. And if you don't remember this, you need to go back to your notes on DNA from last term and make sure that you do remember all of this stuff. It is only when a cell is dividing that that DNA replicates itself, and for a very brief period of time, each chromosome is a double-stranded structure. But under normal circumstances, this does not apply. And so there's no point in expecting to have a double-stranded chromosome when we're dealing with genetics, because a double-stranded chromosome only exists when a cell is about to divide and the DNA has replicated itself. Right, then remember that the chromosomes within a diploid cell, in other words, a cell that is 2N, because it has two sets of chromosomes, and in a human, remember, a diploid cell has 46 chromosomes, but it's not 46 chromosomes in a diploid cell of every organism. So a fruit fly, for example, if I remember correctly, the diploid cell, the chromosome number is 8. Okay, it's only 46 in a human. Okay. Of those, if you remember, because we dealt with this in the section on meiosis, those chromosomes, if they are, a photograph is taken of them and they're sorted according to their length and according to the position of the centromere, I'll get it eventually, okay, and if they stand for banding pattern according to their banding pattern staining. Then remember that those chromosomes, each one of those chromosomes in a nucleus of a somatic cell will have another chromosome in that same nucleus that is exactly the same length, has the centromere in exactly the same position, and has the same banding pattern. And those are the visible things that you can see. Then for every chromosome that is there and is at the same length and has the centromere at the same position and has the same banding pattern, there will be another one within that nucleus that is exactly the same length, has its centromere in exactly the same position, and if stained for banding, would have exactly the same banding pattern. So these two, remember, are called homologous chromosomes. And those two homologous chromosomes, one is always paternally derived, and one is always maternally derived. In other words, this is a copy of a chromosome that was in the egg cell from the mother, and this is a copy of the chromosome that was in the sperm cell from the father. So if you consider a human, and you consider that there are 46 chromosomes per cell, that means that 23 of those must be paternally derived and 23 must be maternally derived. It's not possible by just looking at them to see which one comes from which parent, but they are paternally and maternally derived. Then remember that on those chromosomes there are genes, okay? And so let's say there's a gene here, the third one from the bottom, and a gene here, the third one from the bottom. The genes, the position of the genes, remember, is called the locus of the gene. Okay? So that's the position of the gene. That's just a word for position of the gene. So if you find a gene for a particular characteristic or a particular trait, is the same thing as a characteristic, and the Americans will pronounce it trait, but it's from the French and therefore it should be trait. If it's the same, if you're looking on homologous chromosomes at the gene there and the gene at the same locus on the homolog, then those two genes are always for the same characteristic or trait. Okay? Always for the same characteristic or for the same trait. And those two forms of the gene in an individual, they might say the same thing or they might not say the same thing. In other words, if, for example, one looks at the gene for the ability to roll one's tongue, and I say to you that the gene for the ability to roll one's tongue is dominant, and I hope that it is dominant, eh? I'm just saying if it was. To roll one's tongue is dominant. And the gene to be unable to roll the tongue is recessive. So know those two words, dominant and recessive. Then the symbols that you would choose if you were doing a genetics problem is capital R for being able to roll the tongue and lowercase r for being unable to roll the tongue. Remember, you're not going to choose the letters capital T and lowercase t, because that would mean the individual's got a tongue or the individual doesn't have a tongue. So if you're asked to choose symbols, if they don't give you the symbols, then you always, which one is dominant and which one is recessive, you use the uppercase letter of the dominant one to represent the dominant form of that gene, and the lowercase letter of that same letter to represent the recessive form of that gene. Okay. So if, for example, the allele for where the seeds are smooth or wrinkled in pea plants, the allele for smooth-seeded pea plants is dominant over the allele for wrinkled-seeded pea plants, then that would represent the allele for smooth-seeded pea plants, and this would represent the allele for wrinkled seeded pea plants. So dominant, capital letter, recessive, lowercase letter of that capital letter. Okay. So this dominant and recessive thing, what it means is that if an organism is heterozygous for a characteristic, then the dominant allele will show and the recessive one won't show. So this organism would have smooth seeds. That individual would be able to roll their tongue. Okay. So remember that the terminology you have to know at this point is, if an organism has two alleles the same for a characteristic, then they are homozygous for that characteristic. And you can also say that they're pure breeding, or you can also say that they're true breeding. All of those mean the same thing. The most commonly used term in genetics, though, is it's normally referred to as homozygous. So those two organisms are homozygous for smooth seeds. This one, smooth seeds, that one would be wrinkled seeded pea plant. And then that would be a homozygous individual able to roll the tongue, and this would be a homozygous individual unable to roll the individual's tongue. Okay, so everybody happy with that, I hope. Okay, so in other words, if we're going back to what homologous chromosomes are, then visibly they are the same length, their centromeres at the same position, and if they're stained for banding pattern, they have the same banding pattern as each other. So that's what you would see on them. Not visible, but certainly true of them, is that they always have genes for the same characteristic at the same locus on the two homologs. Okay, so if at that place you find a gene for whether or not the individual is able to roll their tongue, then that will be a gene for whether or not the individual is able to roll their tongue. His or her tongue, which will be there, that's plural. Okay, so that's not visible. And then the other thing that is typical of homologous chromosomes, but you won't see it all the time. You only see it if the chromosomes, if the cell is about to divide, only by meiosis, sorry. Remember that in meiosis 1, homologous chromosomes pair up in late prophase, and they undergo crossing over in very late prophase. And then in metaphase 1, they line up on either side of the equator, opposite the homolog. But you would only see that if the cell was busy dividing by meiosis. You wouldn't see it under normal circumstances. So what you would go on is, are the two chromosomes the same length? Is their centromere in the same position? And if they stand for banding pattern, is their banding pattern the same? Okay. You're happy with homozygous and heterozygous, so those are homozygous and those are heterozygous. You're happy with the term characteristic or trait. You're happy with dominant and recessive. And hopefully you know how to choose the symbols that you're going to use. And then, something came to my head as I did that. Okay, you're happy with paternally and maternally derived chromosomes. Okay, and I think that that was about all that we did on the first lesson. Please make sure that you know your terminology. Please make sure that you can do the genetic crosses that we've done so far. So the students who weren't here today, no, I'm not going to say that now, because then you're going to get all confused about which problems we're on. This takes the place of our first lesson, the one that was held on Tuesday, the 2nd of June. Okay, all right, work hard.